Welcome to Careers in Discovery, your window into the world of biotech leaders. On each episode, we interview the industry's leading executives to understand how they built their careers, what they've learned along the way, and the advice they'd give to their younger selves. I'm back with Martin Brenner, CEO and Chief Scientific Officer of iBio. Martin spent much of the first part of his career in big pharma at Lilly, Pfizer, AstraZeneca and Merck before taking the leap into biotech. We talked about the lessons he'd learned from those early days, the power of simplicity in communication, why leaders need to create space for science to breathe, and how to build R&D teams that are lean, smart and focused on the important things. I'm delighted to be back on Careers in Discovery with Martin Brenner of iBio. Martin, welcome to the show.
Thank you so much for having me today, Tom. Really looking forward to this.
Great to see you, and me too. You just from the surface, just from the research I've done, Martin, it looks like you have a fascinating story, so I'm excited to dive into it. Let's start, though, by what you're up to today. Tell us a bit about iBio, tell us a bit about AI-powered antibodies and the work that you're doing there.
Yeah, iBio is an antibody discovery and therapeutics company. So that means we have a platform that is in part AI enabled. So it's a full stack platform that allows us to discover antibodies, but also optimize those, which is becoming more and more important because, you know, you can create a lot of antibodies that might not have the drug-like properties you want and, you know, be safe and be manufacturable. And so we have built a system actually that combines machine learning, but also biological techniques that are kind of top notch and really novel. And that kind of integration of AI with biological techniques, I think, is kind of where our niche is. Yes. And that allows us to make antibodies that others can't. So we can go after undruggable targets. But we can also obviously do this in a much compacted time frame because we have now shown twice that we can actually go from an idea to a so-called development candidate, which is getting ready for manufacturing and ready for toxicology studies in 7 months. And so that is really fast. And so that is kind of this integration of AI with kind of cutting-edge biological techniques. That's what enables that.
Yeah, it's fascinating, and 7 months is fast. One of the things that's really interesting is, I remember in one of the very early episodes of this podcast, we've been doing this for 6 years now, you were just starting to hear about AI and drug discovery, and it was mostly applied to small molecules because that was where it was easier to train the platforms and things like that. Obviously, antibodies, somewhat more complex, and it's really interesting to see that progression happening.
It is indeed, and you know, it's you saw this kind of huge boom of companies calling themselves AI companies. We steer away from this, right? If you think back 40, 50 years when molecular biology entered the drug discovery space, it changed how we discover medicines today, but basically it did not change a lot of the later stage part, right? So again, we're completely convinced AI will change how we discover and develop medicines, but it's not going to change everything in it. It's going to be a part. Yes. And it's like we're using a computer today. It's completely natural. But, you know, when in the old days, when you actually took notes on paper and took your data down on paper, we walked away from this because it's so much more efficient to do this. So we see this as a tool, not kind of as a, you know, sometimes I get the impression that people feel like there's kind of Terminator style Skynet in the back discovering some drugs. And of course, that's not the case. It's a tool and we can use this to our advantage. We can use this to make medicines we couldn't do before. And I always say making a medicine is at least, if not more than 10,000 steps. And 3 of these steps at iBio are enabled by AI. Right. So it's a tiny amount of different steps, but these are crucial steps. Otherwise, we couldn't make these medicines. And so that's where, you know, we need to be honest. You know, AI is not going to solve everything, but it can be tremendously helpful in what we do and kind of opening new frontiers.
Yes. And I think we've seen this in lots of sectors, including in biotech, is that there is a rush of, and this is not just AI, this is all technologies usually, there's a rush of application to everything. And then there's a step back and go, okay, actually, what are the right things for this to be applied to?
Exactly. And again, every day there's an academic lab or a company that comes out with new models, right? And initially we said we need to create these massive data sets, right? And that required so much computing power. And now some smart people came along and have developed things like partial diffusion, where you need much smaller data sets. And suddenly, maybe that is not the end all, that you need more and more GPUs, more and more computing power, and maybe even bigger and bigger data sets. Maybe it's enough to have, a high dimensional, high quality data set that is much smaller to train a model. So those are the things that are currently emerging. And like you said, at one point, we already have made this. We have made that step back and look, where is it actually working, right? It's kind of silly to apply a technology in an area where it actually doesn't give you any gains. I remember the first lab robots, right? When you wanted to replace humans completely from a laboratory, and you need to move your microtiter well with your cells in there from one machine to the next. And a human being just grabs the plate and puts it on the other one. So basically a second. And the robot took about 45 seconds to a minute to transfer the plate. Not really useful, right? So you have to think where it's really applicable and where you can get gains and can become more efficient.
Yes, absolutely. And I think antibodies are at an interesting point right now. There was a period where everything was antibodies, right? That was the hot topic, and this was the new frontier. And, we had a huge number of breakthrough medicines as a result of that. We sort of then saw the rise of cell and gene therapies, and small molecules made a bit of a resurgence. But I am seeing more antibody companies now coming back to I guess it's sort of probably not even a second wave, a third wave of antibody therapies that are coming through now. Where do you feel the antibody field as a whole is currently?
I think you're absolutely right? We're seeing some really interesting novel developments. I mean, we did have, like gene therapy, like other therapies, had some setbacks, specifically on the bispecific molecules in oncology, right? These could be highly toxic if not dialed in correctly. And we didn't know what we didn't know at the time, right? So we're overcoming this right now with different, T-cell engagers that are coming out. We're coming out with molecules that are masked. So basically antibodies that get activated once they hit a tumor, for example. We're coming out with molecules that hit multiple pathways. Complex diseases usually don't require just one receptor to be blocked or activated. It usually takes kind of more pathways to be blocked and activated. So we can actually now create multi-specific antibodies. One of the biggest frontiers we're working on is targeting GPCR, so G protein coupled receptors. They're really hard to agonize with antibodies. Some of the reasons are just because the steric hindrance. These antibodies are big. There's a big chunks of proteins, and fitting into small binding pockets where usually a peptide or a small molecule binds. This is really hard. So new formats of antibodies like the llama antibodies, shark antibodies, all of those, came out, had their, resistance in the beginning. But if you all put it together, you can actually see where the emerging fields are. And clearly the emerging fields are, smaller formats, high valency, so multi-specific molecules, masked molecules, or conditionally activated antibodies. This is where I think we're going to go. And the beauty of antibodies, that they're precise as a scalpel, right? With small molecules, they get to the liver, and sometimes they get broken down in a very unfortunate way and can cause liver toxicity very late in the game once companies spend a lot of money. That usually doesn't happen with antibodies. So antibodies have different issues. Manufacturing is a lot more complex and expensive. But there's, I think, for every modality we have, there's a reason to be, and there's a disease and an indication and also a target that is better to be, targeted with a modality. So I usually am modality agnostic, but over the past maybe 5 to 7 years, I've fallen in love with antibodies because it goes really quickly to your proof of concept experiment. In small molecules, it can take a year or two until you have actually a tool molecule that actually can be proven that it's working in an animal model. With antibodies, we're there literally in 4 weeks. And so it gives you a much, much quicker solution and proof that this mechanism is actually worth pursuing. And that ultimately gives you more shots on goal. More shots on goal means you're ultimately being more efficient, because you only pursue the targets that are valuable, and you can actually exclude the ones that are not valuable very early on. And again, you cannot solve everything with antibodies, but they definitely have a right to be in this echelon of small molecules, peptide drugs, even protein drugs, cell therapy, gene therapy, antisense oligos. Definitely antibodies play a big role in that space.
No, absolutely. And as we increasingly sort of inevitably move towards more personalized medicine, more precision medicine, of course, a lot of the focus has been on the diagnostic elements of that.
Yep.
However, if you're going to treat in a precision way, then you need precision tools, right? And so antibodies clearly, play that role as well.
Yeah, absolutely.
So tell us a bit about what you're focusing your antibodies on, where the company's at today, and of course you have an interesting dual role at the company.
I do. So we are currently pursuing cardiometabolic diseases and obesity. That's my background as a scientist. I started that way in my career many years back at Eli Lilly, and somehow got into rare diseases over time, and then into oncology, and somehow I snapped back and came back to cardiometabolic. And I feel still at home.
Yes.
The problem is really that a lot is known, and there's not a lot that has been done on novel targets. Quite a little bit, of course, but it's a massive field, and it takes years and years. I still don't consider myself an expert because there's still thousands of papers I should be reading and haven't read yet. So it's a massive field. But we're in this space. We obviously pivoted in this area last year in March. And so we first sat down and thought about, so how do we tackle obesity, cardiometabolic? As you know, Lilly and Novo Nordisk, two major companies out there that have been doing peptide drugs for the better part of a quarter century. It felt silly for us to kind of compete with those giants in that space. So we decided we're not going to go into the GLP-1s. And there's also hundreds of entrants into that space.
Yes.
So how do you actually differentiate yourself? So we came clearly out of this thought exercise saying we want to treat on top of GLP-1s or after people rotate off of them, or we want to provide alternatives. That was the first kind of pillar of our strategy, if you will. This next one is, and again, I'm discovery scientist at heart, so I always want to have novel targets, but novel targets carry a lot of risk as well. So we always balance the portfolio. Our portfolio right now is really balanced towards validated targets, and there's multiple definitions how you consider a target validated. Genetic validation is the simplest one, right? You see a loss of function in a molecule, and it's related to a disease, and you agonize the target, and you see a benefit. Or you see that one variant is causing a disease, and you knock that variant down, or antagonize that variant. And so specifically in the obesity space, there's a lot of these targets. Like myostatin, we're working on a long-acting myostatin program that's differentiated from all of the molecules that are out there to basically retain muscle mass.
Yes.
Unfortunately, when we lose a lot of weight really rapidly, we not only lose fat, which is the desired part of the body we want to lose, we also lose bone density and muscle. And this loss in muscle, we believe, can be really, really detrimental in specifically with people getting older, where you anyway look at sarcopenia, loss of muscle over time. And so we want to counter this. So myostatin, there's multiple people on the planet walking around with a loss of function of myostatin, and the only thing they have is they're very strong, very muscular and lean, but no other adverse effects on their health. So we feel it's a safe mechanism, but it's also genetically validated. So if we block this, there's a pretty good chance that we actually see this increase in strength and in muscle mass. Another one is activin E, which is a really cool target. So two large companies, Regeneron and Alnylam, have actually linked this target to obesity, cardiovascular disease, and diabetes. And so we were wondering for a long time why Regeneron, who's kind of a really, really well-known company for making antibodies, didn't come out with an antibody against the target. Well, we figured that out because this protein is really hard to express outside of the body, and it does not fold correctly. And antibodies actually recognize surfaces, not amino acid sequences. So if the surface is different, you can create antibodies, but they don't pick up basically the endogenous protein. And using our AI-enabled discovery platform, we were able to kind of work around this. And so we're very proud to be the first to kind of have this. But that's a really cool target, because at the moment we think we have to lose a quarter or 25% or 25 kilograms of body weight to become lean. But if half of that is muscle, that's actually not good, right? What we want to lose is fat. And activin E is really interesting because what we've at least shown in preclinical models together with competitors that knock the gene down in the liver is that you cause a fat-specific weight loss. And so you only lose fat. And we could show that you only have to lose about 5% body weight to lose a quarter of the fat mass in your body. And so this is really intriguing and novel as well. So this could actually kind of solve a lot of issues with bone density loss, with loss of muscle mass that we see with other weight loss drugs. So we're really excited about those two targets. And they basically fit into the third part of our strategy, because it's really hard for others to make them, because we can use our technology to make them and the skill of our team. And that kind of creates a barrier, because there's hundreds of companies starting in the obesity space, well-funded. But again, we're happy to take any of the competition, because ultimately you need precision, you need speed, and you need high quality molecules that actually make it through the clinic. And we're one of the few companies that can provide all of that.
Yeah, absolutely. And I think this is a really interesting space at the moment, because you kind of referenced it there. Perhaps with the exception of cancer, there's a huge amount of historical data around cardiovascular and metabolic diseases, right? It's probably one of the most established fields. However, the kind of nature of the issues is changing. Maybe the diseases haven't changed, but we used to have a lot of heart disease, for example, because we drank gallons of whiskey and ate fatty food and smoked and all this kind of stuff. Now it's more about obesity and sugar and these sorts of things. And I suppose some of it must be about, well, yes, we have, whereas if you're in rare diseases, as you talked about, it's kind of, we don't know much about this and there's not much data. Now there's this wealth of data, and sorting the signal from all of that is, as you said, there's thousands and thousands of papers you could be reading, right? And you'd be reading them forever. But it must be interesting to sort of take that data, find the signal in the noise, as they say, but also then adapt it to, okay, well, the nature of the problem has changed slightly.
Exactly. I mean, if I was actually in a lab at Lilly when the first GLP-1s were coming out, and we tested them for insulin release, that actually was my PhD thesis, to find small molecules that would mimic the action of GLP-1. And so it was very interesting at the time. Nobody was interested in obesity. And we saw some of these molecules really reduced body weight. And at the time, the sentiment was obesity is not a disease. Type 2 diabetes is a disease. Obesity just leads to it. And so there was always this thing, it's basically a pre-form of the real disease. We shouldn't treat that. And of course, as we all know, the best protection against disease is prevention. And so it's sometimes really hard to convince people, do the right thing to your body, be responsible, so you actually won't have all of these diseases. We made great strides with, the lipid lowering agents to kind of prevent, these full-blown heart attacks. We now have other issues. We have smaller heart attacks that lead ultimately to heart failure. So there's another disease that's coming up. But obesity really has not been in focus as a disease, almost disregarded. And, I think it started, honestly, like many products, with the demand of a consumer, right? It's probably a few people here in California who said, I want this for weight loss. And that kind of started spiraling. And suddenly it has become actually, considered a disease. We always felt this is really kind of the crux, right? We do have a problem with energy intake. We eat way too much. We eat way too energy-dense foods compared with 50, 60, 70 years ago. And we don't exercise enough, right? And if you think about it, right, our bodies are trained to store energy and release the energy. If we keep storing energy, something will go wrong. If we keep spending energy and not refueling, we know something will go wrong, right? If I'm an endurance cyclist, so I know exactly what happens if you don't fuel. You sit at the side of the road and there's no way to go forward. But it is really important to kind of keep this. It's called metabolic flexibility, was coined, I believe, at UPenn at the time. And we need to kind of keep that flexibility for us to stay healthy. And if you constantly load up on calories without expending them, this is not a good idea, right? And again, what we're trying to, or what people tried in the past is, create, and we call it exercise in a pill. That backfired massively because, again, this is, you cannot replace exercise. If you move your muscles, many things get influenced. You cannot actually do this with a single pill. And unfortunately, there's no way around, but we have to move. We're humans, we're made that way, and we're working basically against our nature if we don't. And so that's something we cannot tackle. What we can tackle is, there's increased energy intake. We can reduce that, but it's not like smoking. You cannot stop eating. You're going to be malnutritioned. And, there's some voices saying, in these late stage clinical trials of these drugs that reduce body weight by, 25% and more, people are malnutritioned, right? So what's the opposite? The opposite is to kind of increase your energy expenditure, because guess what? Our bodies can actually go into energy preservation. Again, feast famine in the Middle Ages in Europe, that's what our bodies got trained for, right? And that's what the selection criteria was. Gain quick, so you have actually something in case of a famine. And so basically what this means is that if we can actually increase energy expenditure, right, consume more energy while we're resting, for example, that would be a great addition to, reducing food intake. That would be actually perfect. Now, this is really tricky, and there were drugs in the 1920s, highly toxic, killed a lot of people, but you can uncouple your mitochondria. And if you uncouple your mitochondria, you're just burning energy. It's like you take the gear out of your car and hit the gas pedal, and you're basically revving the engine up. It will consume gas, but you're not going anywhere. That's basically what uncoupling is. So you're burning energy, and so that can be useful, but it can also be dangerous, right? It can overheat your tissues. It can, cause all kinds of other issues. It can lead to respiratory arrest and all kinds of really nasty things. But there's better ways of doing this. For example, so imagine there's a sedentary, overweight person and an athlete, and you put them on a sofa and let them rest for a full day. Guess who burns more energy? It's the athlete that carries a lot more muscle, because muscle has a lot more mitochondria, and to maintain muscle, you need more energy, right? Fat is a pretty inert tissue, and it's made that way. It's supposed to be a storage container. Well, I'm going to get killed for this because, of course, it's also a tissue that is secreting hormones and doing other things. But one big part of adipose is to store energy, right? And so muscles are made to kind of consume energy. And so if we build more muscle, and if we build those muscles out to be fully functional adult muscles, not just, embryonic, early differentiated muscles, we will definitely see a benefit on this energy expenditure, which then will be healthy versus, uncoupling your mitochondria, which basically leads to no, physical movement or anything, just to kind of heat generation, if you will.
Yes. Yeah, absolutely. Do you know what? It's fascinating, and it's also really interesting as you're talking about that. How many of these conversations I have with people where they say something along the lines of, I worked on something like this in my PhD, and then I didn't work on it for 20 years, and that's now what I'm doing. And it's amazing how how often it comes around. But I do want to take things back, Martin, to the beginning of your career, because you didn't start down this path, from what I can tell. You trained as an electrical engineer originally, I believe.
Yes, that is right. My entire family is leaning towards engineering. My dad's an electrical engineer. My uncle's a mechanical engineer. My grandpa on my mother's side was a car mechanic with his own shop, which I spent a lot of time in. My grandpa on my father's side was an architect. So everything was more on the technical side, right? And so my parents never pushed me in that direction. They actually offered me a lot of different things, right? Language, travel, and I wasn't pushed one direction or another. But naturally, my dad's my hero, so you fall into that. And I wanted to become an electrical engineer because he was an electrical engineer, and he's retired now, of course. But so this is how I started down that path. And I was not good at school. I wasn't focused until I met my wife, who kind of snapped me into focus, if you will. And then I realized, I I could become an engineer, but I would never be a brilliant engineer. I would always be kind of an okay or mediocre engineer. And at one point in your life, you have to think about what do you want to do. It is very difficult for me to think that, you're spending your entire life not doing anything meaningful. And so I wanted to do something where I'm really good at, where I can actually make a difference. And so I did a lot of soul searching, right? It was not an easy decision to drop out of electrical engineering after 3 years, actually. So that was a hard decision to make. And so
Yes.
I did a lot of soul searching. And ultimately I said, okay, before I jump into this, I need to get some practice. I need to see what the job really looks like. And among the many things I tried, I actually, volunteered in a veterinary clinic. And that brought together two things, and I didn't realize it at the time, but it brought two things together that today still fascinate me. It brought in engineering, and it brought also in really solving tricky things on the biology side. And engineering was, the clinic actually was doing a lot of really complex surgeries on companion animals. So putting a platinum plate on a femur of a dog is actually quite mechanical. This is quite an engineering feat. So that brought that in, and I felt like, I get that, I understand this. But then having actually complex problems to solve, diagnose animals that come in and try to find out what actually you can do to help them, this was fantastic. I loved this, right? And so I immediately felt like after 6 weeks in volunteering in this clinic, I thought that's it for me. So I applied, and with my grades from high school, there was no way I could get into veterinary school. But luckily, there was one exemption, right? You could do a special test that it's now obsolete and people don't do that anymore, but it helped, understand your three-dimensional vision, your logic, your reasoning skills, and so on. And if you made it to the top 5% in the country, you would be considered for getting a place. And so luckily I got to the top 5%, got in and studied veterinary medicine in Munich, in Germany. And I loved it. It was tough because we couldn't afford living in Munich. We lived in Ulm, which is 100 miles north of Munich. And so it was a burden. We left every day at 5:45 in the morning with a train to Munich, then hopped on the subway and walked down to the veterinary medicine faculty every single day for 6 years. And so the first 4 years it was helpful. I could study on the train, but then it really became a time-consuming issue. And again, it was pretty bad at the end, but I made it through. And on the way, I was really gearing up to become a small animal surgeon. I have pretty decent, surgical skills. But in about 4 years in, I met my professor for pharmacology. His name was Petter, Professor Petter. He was retired and brought back because, there was a shortage in professors. And he was one of these old school pharmacologists who literally tried the drugs he prescribed to his patients before he actually prescribed them. And so his classes were so colorful and so immersive. I felt like, wow, right? I want to make medicines. And he actually pushed me down that path, and not knowing even. And so I thanked him years and years later. He unfortunately passed away many years ago already, but I thanked him afterwards because it really was kind of the change in my career that I needed. And again, I felt like, I'm changing fields again, but it felt just like the right way. Everything fell into place at that time, and started my PhD in that space. Actually, in Germany, you can do a PhD. You can split it. You can do your coursework at university, and then you can do the practical stuff at a company. So at the time, the biotech sector in Germany was meh. 1999, 2000 was nothing.
Yes.
And I sent about 100, a little bit over 100 resumes, and it was all still paper-based, right? So print it out, put into nice envelopes, send it out to companies, and I got literally 2 responses. Okay. And this is one of the big learnings, right? Your resilience will come in really handy. And one of the responses was from Eli Lilly, and they had just started in northern Germany with a research site, brand new. And one of the VPs on that side was an old school pharmacologist, and he liked veterinarians because he said, well, you have a very diverse, background. You can do a lot of different things, and I have a problem we couldn't solve here, and I want you to solve that problem. And that problem was he had a molecule that worked like a GLP-1 peptide, a small molecule, but they could not develop it because it always showed toxicity. And so he brought me in as part of my PhD thesis to kind of assess which of these molecules from that series they discovered could potentially become a drug. And so that's how I got hooked on this. So I got almost instantaneously access to drug discovery programs at Lilly, was integrated in their drug discovery teams. And that's exactly what I wanted, right? I wanted the science to be applied and not for the sake of science. Don't get me wrong, there's a huge and urgent need to have academic science. It was just not for me. And so for me, that was the perfect situation.
No, that makes sense. And I just want to highlight what you said there about resilience, because I think we're in a similar situation for a lot of people right now, right? The job market is not great in the biotech sector, and there are companies hiring, but not a huge amount. And it's interesting because at the beginning of the year, we ran a number of workshops for people who were looking for work and finding it difficult. And we were talking to them about, how to run a job search and how to be effective with that. And in every single one of those workshops, I answered a variation of the same question. Someone would say something like, I've applied to loads and loads of jobs and I'm not getting any response. What am I doing wrong? And I would always ask, how many jobs have you applied for? And invariably the answer would be somewhere between 3 and 8. And I'd say that's what you're doing wrong. But it's difficult, isn't it? Because people people have this perception that, well, I need to take my time with each application, and if they don't get back to me, then I've done something wrong. And I don't think that's the case. I mean, you identified it there. You made 100 applications and heard from 2. I don't think that's that unusual a ratio, actually, especially in a time like this.
Yeah, 100%, right. And I try to mentor 4 to 5, young scientists that they want to get into this industry. And the first thing you always hear is, I never get beyond the first interview, right? And so, yes, this is perfectly fine, right? This is, you need a practice. You don't take up a guitar and suddenly play like Eric Clapton. It's not going to happen, right? Sorry, you have to practice, right? And so first of all, you need to get through the HR interview, right? Because that's the screening part of it, right? There's hundreds of applicants. How do you actually get through this, right? And again, like we said before, biotech has its own language, its own rules. And how do you actually how can you peek behind the curtain to learn these rules while you apply? It's really hard for the first job. The first job is really just get the first job, do a good thing, and then move on to the job you really want, right? So you need to get that first job. It's really, really rare, exceedingly rare that your first job is exactly what you wanted, but it gives you the entrance. It gives you a chance to prove yourself, gives you a chance to understand how things work, and then the second time is going to be a lot easier. Yes. For me, when I joined Pfizer, after I spent almost 10 years at Lilly, Lilly was like a family for me. My colleagues in Germany and in the US, I relocated to the US, halfway through my stint. It was the hardest thing for me to leave Lilly. But at one point, your career trajectory does not match with, what the company can provide. And then you have to make these decisions. And going to Pfizer, I knew, more who I was, right? So again, I also want to highlight, when we're not perfect human beings and we need to learn. If you stop learning, you're done. Your career is done. Yes. I still to this day try to learn one new thing every single day. And I just make it a habit to get better at what you do. And so again, I had many times in my career where it looked bleak, where, I felt like I'm not going forward. Listen to mentors, to advice, adapt to new situations, learn to do new things. Don't take a job that you can already do with one hand on your back. Take a job that scares you a little bit, right? That has parts in it that you're worried that you can't do right now. That's it's like exercise, right? If you don't push the system, you're not going to have any gains, endurance or strength-wise. And it's the same with the job. Stretch yourself, right? Be confident that, hey, I don't know everything yet. Be honest about this, but also say, but I'm going to learn it, right? And this learning part, this was to me always important. Every single job I started, I was not really good at that, but I became good at it. I learned something from it, right? And the next job, I could apply these learnings. But if you basically stagnate and don't challenge yourself, you're leveling out your career. So take risks, right? I mean, when the best time to take risks is now. It's a universal truth. You're not looking back at your life and saying, I'm so sad that I did these things. Usually you're saying, I'm so sad I didn't do something, right? And I think this is often where people get stuck, right? I have a lot of friends still in big pharma, and they see biotech life, and biotech life can be really, really good. It can be really hard. There's no nice hotel rooms. There's no business class flights. It is hard because you're focused on efficiency and focused on one thing only, making medicines. But to do that, you have to actually change and be willing to kind of sacrifice certain things. And so a lot of my large pharma colleagues, and when I talk about biotech life, I always say, if somebody needs a job, work with us for a week. And if you still want to be in biotech, I'm happy to discuss this. But usually after a day, people realize this is not for me, right? And again, this is also a big question of where do you want to end up, right? Do you want to focus your career on large pharma? Do you want to focus it on biotechs? I feel you should explore both, right? You might like something that you don't even know you like, right? Large pharma offers you a lot of opportunities, learning, growth. You can look into different areas, right? You can learn about sales, about manufacturing, about all these things that in a small company you're usually not exposed. If you want to be part of a drug discovery team that goes all the way from idea to maybe approved drug, a small company is much better because you see more, you have more responsibilities. But each opportunity, each company with different sizes offers different opportunities, and I can only highly recommend to do both and figure out what you like best. And maybe it depends on the stage of your life. I'm at the moment I feel very, very good about biotech. I actually would feel quite limited in large pharma right now, but that was not always the case. When I was learning and growing up in science organizations, it was great to always have somebody to ask. Now it's scary to not have anybody to ask anymore. So you need to have a mentor network where you can call somebody and say, I just ran into an issue today I never ran into before. Who do you ask, right? In a large company, there's always somebody to ask. If you're the CSO or CEO in biotech, there's nobody. You're the one who solves this, right? And that's why you need to establish these networks. It's really important. They're important for knowledge exchange, but they're also important, like you said, right? After the 10th application, if you get a no, sometimes you feel like, is it me? And it's not you. I can guarantee you it's never the person who applies. It's just finding the right job for the right skills. And that might take some people more time and other people less time. And so that is really important, that if you're down, that's why you need a team. That's why this is a team sport. You can fall in a hole and be desperate, but not the entire team at the same time. One person at a time is fine. And then you have your teammates to pick you up and help you out. And this is how really, really successful teams work, right? This is why you need your team around you, because it's going to get hard. One thing I can promise for anybody who gets into biotech, there's going to be hard times. And science usually kicks your butt one way or the other, because the next experiment could kill your program, which happens all the time. And it might be your most beloved program that has the most promise. And, one experiment that you design, that you have to design to prove it's safe, it's doing XYZ, could be actually the end of it. And those are really bad times, right? We're not making stamps, or we're not making, a stationery. We're making medicines that, hopefully will help people. And so that's why it's so devastating to kind of fall into this. The first 10 programs I led all died before we actually went into tox studies. So it's also, it can happen, right? It was probably a little unfortunate, but those are the things that will happen, and you need help. I barely know anybody who actually sustains these constant blows. I always say, you're going to get sucker punched once a day. And you need people around you that have gone through the same and can lift you back up. Yeah, it's a really interesting point, and I think there's a few things in there that I wanted to sort of draw out. The first is that there's sort of been this trend. I don't know, maybe it's starting to shift now, but there's been this trend over the last few years of people
feeling that working for a big company isn't as good as working for a small company, or less exciting, or that kind of stuff. But there's some, and it depends on the person, it depends on their sort of way of working and their preferences. But there's some real advantages to seeing what world class really looks like, and sort of what the level really is. And then I think a lot of the time what happens is that some people thrive in that environment, and then some people kind of you hit a point in your career, which probably, looking at the roles that you held and the seniority of roles that you held, you're probably hitting this point where it takes a long time to progress, right? And you get into some congestion, and then that's where some people move. But I think, as well, as you say, you know, the failure rate is so high that it's easy to get discouraged, and that prevents people from taking risks. And I think there's different types of risks, right? There's the scientific risks that you need to take, but there's also, you talked about career risks there. And I've seen a lot of people who actually didn't take a job because it was too much risk. Yeah. And every time it happens, I know that person's going to regret it, with a few exceptions. Yeah. A couple of them were good decisions, actually. But it's a really important point, is like, I think you and I are probably quite aligned on this. I would much rather do something and regret it than not do something and regret it.
100%. Exactly. Yeah. There's always, you always make, even if you make the wrong choice for a new job, you can always correct it, right? Mm-hmm. You can always say, we're not prisoners. We're free. And free adults, we can actually choose where we work. We don't have to, right?
Yes.
And so trying things out is really important. This is how, you know, really big inventions happened, right? The transistor didn't come along because somebody thought about this from the start, this would happen. They played around with the different components until they actually found something, right? So you have to also try, right? Now, again, there's things, if you try and it didn't work and you try again the same thing and it doesn't work, well, you know, that's you need to learn from that, right? So and again, I learned over the years to you know, don't get cocky, just stay humble, because this is a much bigger problem that you're trying to solve, right? And especially when you're working with people, I tried so hard to kind of be a good judge of character, try to kind of figure out what is the, you know, the C-suite of a company, how are they made up, how do they operate, is it on the ethical level where I want to be, all of these things. And I still, you know, ended up in situations where I thought, how could I not see this? And it's important then to reflect back on this and say, okay, what did I actually miss? And again, this is a scientific principle, right? Do an after action review, if you want to take a military term for it, right? But after battle, you know, make sure you go back and say, hey, what did we do well and what could have gone horribly wrong, or what has gone horribly wrong, right? And so I think this is really important. So I can highly recommend to everybody listening, once a year, take a retreat, take a few days. I usually go in the desert here in Southern California, which is great. You're all by yourself. You have the stars above, you know, you, and there's quiet, and then reflect on the things that actually worked and didn't work. And this is so important to kind of get in the right mindset for your next decision. Don't make rash decisions, right? I'm always trying to come up with kind of pros and cons for making a decision versus another one. But once that balances out, it's going to be a gut decision. And those gut decisions are very often right, but they can also guide you horribly wrong. So this is really important to understand, is what emotions are driving your decisions, right? We're all emotional at times. And I'm not saying you should make a decision without emotion. Getting into biotech is an emotional decision, right? If you actually think about it, right, from a perspective, it takes 10 years to make a product. The failure rate is 90+%. Why don't you make cars or anything else, right? This is much safer when it comes to being successful. If people are driven, and again, we're all driven by certain things, you know, it could be money, it could be power, it could be just, you know, influence and a respect for your name, whatever drives you. Biotech is usually not scoring highest on that list, right? If you're after money, go run a hedge fund. This is so much. I'm not saying hedge fund running is easy, but to get to be a rich person is so much easier in other fields, right? We're not in for the money. And this is what I often see in social media, the bad big pharma, and they're all out for money. We all went in there knowing that, we're likely would have made a lot more money anywhere, any place else. This is honestly an emotional decision to do this, right? Making medicines is a passion. And anybody who thinks we're in for the money has not ever worked a single day in biotech. And I encourage everybody who thinks that to please join us, right? And usually they run with their hair on fire out of the door after an hour because it is hard, right? And it's a passion thing for many of us. And we wouldn't be doing it if we didn't feel like this is necessary and we do something that is meaningful.
Yes. Yeah, and I 100% concur. I've worked in this sector for nearly 20 years now and spoken to tens of thousands of people in biotech and pharma about their careers, and all of them, obviously people have stuff going on in their life and you have other motivations and things happen and all that kind of stuff. All of them fundamentally do it because they want to have an impact. They want to make a difference. They want to help people to feel better, live longer, whatever it is they're focused on. I can count the exceptions on one hand, honestly. And it's something, as you say, that people outside the sector don't realize. I just wanted to ask you about that decision that you made to move into biotech as opposed to big pharma, because you spent, you'd spent the first 16 years of your career at some of the biggest companies in the sector. And I imagine when you make that decision, there's probably mixed feelings around risk, around what it looks like, all that kind of thing. Just could you tell me a little bit about how that decision came about? Yeah.
I want to touch on risk first, because I have seen more change in my 3 or 4 years at Pfizer than I have seen in any biotech company I was at. It's just not the entire company, it's parts of it, right? There's constant change, right? So big pharma has a perceived safety, right? It is not. It is by far not kind of unchanged, and it shouldn't be, right? Change is really good for innovation. It's very uncomfortable sometimes, but it's good for innovation. So for me, being in big pharma for 15 years, I've seen a lot of different company cultures, right? Lilly was very family oriented when I was at Lilly. Obviously, it was a long time ago. I couldn't and wouldn't want to judge what Lilly is doing today. Jumping to Pfizer was way more kind of a really organized, plan every step out organization where everything is on purpose, right? That was a really, really important learning for me. It had downsides as well, right? When it came to treating people, specifically in times of change, there were things that I wouldn't agree with, and how people were treated. But that's just personal style. Going to AstraZeneca was really interesting because AstraZeneca was, in the old days, Astra was one of the leading innovative biotechs in the world. And when I joined, it was kind of reduced to a Me Too organization. So they did only things that others already had done to de-risk everything. And the scientists, and they had brilliant scientists with, science cover papers that were buried under layers and layers of bureaucracy. And for me, kind of removing that bureaucracy and liberating the scientists, putting them into self-organized, if you will, small teams to operate semi-autonomously so that they felt like they were empowered to do what they needed to do. That was really important for me. It was very controversial at the time at AstraZeneca, and a lot of people didn't like it, but the few scientists that were really brilliant, I think they really benefited from it. And it was changing the culture. And then coming to Merck in the US, the goal for me was to build a biotech within a large organization with the thought of getting the best of both worlds, right? Building a biotech, 15 people, and I hired really, really brilliant people. I was very, very lucky to get kind of the golden generation from some of the key labs in the United States. Really brilliant people. And I think we scared almost half of the rest of the organization with the speed and efficiency we were operating, and that caused friction, right? And like you said before, you need different skills in biotech and in pharma. In pharma, it's a lot of, if you will, diplomacy. You need to kind of navigate and negotiate your way through, because there's many, many different organizations. You need to actually get them all aligned behind your goals, and that requires about 80% of your time. and 20% are left for your team and for science. So there's not a lot of time for science left.
Right.
And at the time, obviously, what kind of, I shouldn't say broke me, but what kind of made the decision for me was that the misalignment within Merck.
Yes.
So we basically had broken our backs, my team and I, to kind of bring an antibody forward in a heart failure indication. And when it came to clinical development, we were basically told, before we actually put this molecule in the clinic, we would rather do another trial with Merck's cancer drug Keytruda. Now, strategically, absolutely the right decision for the head of R&D, right? No question about this. For us in the trenches, trying to actually bring other medicines forward, telling us after that time of trying and moving this molecule forward, we're not going to do this. There's this misalignment and this kind of, changing and conflicting interests of different groups, that was it for me. I felt this is more hindrance of making medicines than actually helping me, right? And again, what helps a lot is that you have a fantastic clinical team that can actually move through clinical development really fast. You have a fantastic team that makes your molecule in quantities that you can actually then use in animal models. So you have literally everything at your fingertips, but this misalignment of strategy, if you will, across an organization, that was something I felt like this is getting highly political, and I obviously can navigate this. It's not my favorite because again, it prevents me from making medicines, but I wouldn't have gotten to where I am if I wasn't good at that. But the problem was really at this point in my life, I felt like this is not helping me make a medicine anymore, right? And this is when I jumped into biotech. I felt I just had built a biotech within the walls of Merck. Why not go out in the real world and do it myself? And that's how I ended up at employee number 8 at my first biotech company. And I stepped into this place, and this was the second time in my life I felt at the right place at the right time.
Right, I see.
It just everything fell in place.
Yes, and then we're fast forwarding quite a bit from there, but over then that time, you've worked for several biotech companies and been in leadership positions in those and executive positions. What do you think, if there were maybe one or two things you've learned about the biotech end of the sector that would have been good to know at the beginning? What do you think of those kind of one or two key things?
So there's a ground truth that is true for all biotechs. Biotechs have one advantage over pharma, and that is speed, because the entire team is basically telepathically aligned, right? This is why you do this. You spend 16, 18 hours a day with your team. You have one goal and one goal only. And this alignment is what creates the speed, right? There's no question, should we run program X, Y, or C? How do we prioritize this? You have your priorities set.
Right.
And this alignment, this is something that needs to be negotiated really hard in a large company, and that is basically built into a small company. That is, in my eyes, the biggest difference, right?
Yes.
And this is when you see biotech companies starting with, $800 million or $1 billion, you're literally shooting yourself in the foot, because then they're actually forced to build really rapidly, which slows everything down. This is completely going against the purpose of a biotech company, right? It is not capital efficient. It is not fast anymore. You have to now manage $1 billion, and your investors want you to spend that money real fast to create value. And so it really defies the purpose, right? And so we've seen this now a couple of times. And again, this is a nice way of parking a lot of money, because you're getting probably a 2x return. But biotech was always built on 10x returns. So speed definitely is something that, and I'm not saying this is not possible in a large organization. You just have to hire the right people, right?
Yes.
If you start going down the path and a single mishire that is not corrected can actually destroy an entire team. And I think this is the risk in pharma. It's just very, very risky if you start building to have that one mishire that then can actually start building, their silos, and then from that point on, it's really almost a point of no return.
Yeah, that makes sense. Well, Martin, there's so much more we could talk about. We are out of time almost, unfortunately, but there's so much more we could dig into. Just tell us a little bit, I suppose, to wrap up about what's next. What's on the horizon for you and iBio, and what are you looking forward to?
So we're looking at the biggest milestone any one of us always has. We have our first molecule, our long-acting anti-myostatin, hopefully being dosed to the first human being next year, in the first half of next year. That is for us the biggest, milestone. To see that all of the work actually ended in, a molecule that actually is safe to treat human beings. That this is the biggest milestone. So that's what we're looking for right now. Obviously, the markets are really, really horrible right now. And, insecurity worldwide is not a good thing for biotech. But we feel like we're really well positioned. As I said, we see this competition from China coming with a lot of biotechs being efficient. We're happy to take this competition any day. We have the precision with our anti-AI-enabled discovery platform. We have the speed, but we also have the quality molecules that come out of this. So I think we're one of the few companies that have a platform, but also have a well-crafted obesity strategy. So we're really excited. And we hope that our first molecule will hit the clinic very soon.
Fantastic. Well, it's a big moment when it does, and we wish you the very best of luck with it. Thank you so much for joining us, Martin.
Thank you so much for having me, Tom.
Thanks for listening. If you enjoyed the episode, please like and subscribe and share it with someone else you think will benefit. And for more podcasts, videos, blogs, and insights, come follow me, Tom Froggatt, on LinkedIn, YouTube, Instagram, Medium, or TikTok. See you next time.